Hello, and welcome to today's webinar hosted by LabRoots and produced by Taconic Biosciences. Our presentation today is titled Characterization of Myeloid Cell Hyperactivation Syndrome and Survival Differences in Humanized NOG-EXL and NSG-SGM3 mice, presented by Dr. Elinor Willis. My name is Dr. Caroline Mitchell, and I'll be your moderator today. I work closely with Taconic scientists and researchers in academia, biotechs, pharma, and beyond to develop scientific and educational content related to preclinical mouse and rat models. Let me now introduce our speaker. Dr. Elinor Willis received her veterinary degree and PhD from the University of Pennsylvania. Her PhD work focused on antiviral immunity and vaccines in infant mouse models. She then completed her residency in veterinary anatomic pathology at the University of Pennsylvania. She is a diplomate of the American College of Veterinary Pathologists. She is currently a fellow with the Comparative Pathology Core at UPenn, with interests in animal models, immunology, cellular, and antibody-mediated therapies, and infectious diseases. So let's get started. Elinor, please begin when you're ready.
Okay. Thank you everyone for being here. I'm just going to turn my camera off during the presentation. Okay, thanks for joining today to hear about myeloid cell hyperactivation syndrome in NOG-EXL and NSG-SGM3 mice. At the beginning, I'm going to talk a little bit about the important mouse strains involved in this work, and then we will get into the details about myeloid cell hyperactivation syndrome. But just to start off, you are all probably familiar with this, but I just wanted to talk a little bit about why humanized mouse models are important and useful. I've made a list here. I'm sure there are others, but I just wanted to point these out, and especially the fact that a lot of these have to do with the immune and hematopoietic systems. And then in addition to thinking about why we use humanized mouse models, it's also important to think about how they're made in the context of the rest of this talk. So you need two ingredients for making a humanized mouse model. The first is the host mouse strain. And there's a range of them that can be used, starting off with nude mice. So these are deficient in T cells. Skid mice, which are deficient in T and B cells. Not skid mice, which add an NK cell deficiency to those T and B cells. And then NSG, NOG, and NRG mice, these are all similar, and they are deficient not only in TB and NK cells, but also complement and histiocytes. And by histiocytes, I mean dendritic cells in the monocyte macrophage lineage. And then really importantly for this talk, NSG, SGM3, and NOG-EXL mice. So these strains are both based on an NSG or NOG background. So they have those immunodeficiencies, and then they additionally express human myeloid-stimulatory cytokines. Both strains express GM-CSF and IL-2, and then the NSG-SGM3 mice also express KIT ligand. And these mice were developed to have improved myeloid engraftment. All of these mice, whichever strain you're using, are typically preconditioned, and then the second ingredient, the human cells, are added to the mice. And these might be peripheral blood mononuclear cells, peripheral blood lymphocytes, hematopoietic stem cells, or a bone marrow liver thymus model, for example. And so thinking about our humanized mouse models, if we think specifically about the myeloid supportive mouse models, these are particularly helpful for some of these areas here. And again, I want to point out that a lot of these have to do with the immune system and its interaction with infectious agents, tumors, transplanted tissue, things like that, and they can be really important for working on approaches to personalized medicine as well. But as a lot of times in science, sometimes you take 2 steps forward and 1 step back. And while these mice are really useful, these mouse models are really useful for studying those areas that I talked about, they have some limitations as well. And one of those is myeloid cell hyperactivation syndrome. And this can lead to increased morbidity and mortality of the mice. And this is detrimental to the study, obviously, and to the mice themselves. So myeloid cell hyperactivation syndrome has been called, or features of it have been called, macrophage activation syndrome and or hemophagocytic lymphohistiocytosis, or MAS or HLS-like disease in mice. It also has components of mast cell and eosinophil hyperplasia, as shown in the title of this paper here. So in our study, we use the term myeloid cell hyperactivation syndrome to unify these different components, the macrophages, the mast cells, the eosinophils. These are all myeloid cells. So we use the term myeloid cell hyperactivation syndrome more generally. This syndrome has been described in NSG-SGM3 mice, also called NSG-S mice, in these papers here, and the pathology has been nicely described in these 2 papers here. It's also been described in NRG-SGM3 mice. Parts of the syndrome have been reported in NSG mice. We recently published a paper in which we characterized the syndrome in NOG-EXL mice because that hadn't been previously reported in the literature. And so I'm going to focus on that from now on in this talk. And we compared it to the NSG-SGM3 mice because it was better understood in those mice already. As I get started telling you about our study, I want to give you some context. So the goal of this study came from the investigator we were working with. So the Comparative Pathology Core, we're a group of pathologists, and we work with investigators across the university and at other universities and companies as well. And so we were working with an MD clinician scientist who works with glioblastoma multiforme patients. And his goal was to develop an autologous model in which the patient sample and the patient's own immune cells could be studied together. In particular, he was interested in the intratumoral myeloid-derived suppressor cells and regulatory T cells. And so the plan, the long-term goal, was he would identify patients who were undergoing surgery for their glioblastoma multiforme, and then those surgical samples would be cultured into patient-derived xenograft organoids, and then those would be implanted into the mouse model. And in the meantime, those mice would also receive hematopoietic stem cells from that same patient. And so because this investigator was particularly interested in some of the myeloid cells, a myeloid supportive model seemed like a good choice for him. And so the Comparative Pathology Core did some previous work with him that was published in one of those papers I showed you on an earlier slide, investigating whether the NSG-SGM3 mouse would be a good model. And they ran into some problems with the myeloid cell hyperactivation syndrome. So now in this study, we wanted to see whether the NOG-EXL mice would be a better option for this study and other studies like it, where the myeloid cells are a really important component. And in studies like this, where you need a long window of time between humanizing the mice and the studies that you're planning to do with it. So I'll show you the more granular details of our experimental design. We had 3 groups of humanized mice and then also non-humanized controls. One group consisted of NSG-SGM3 mice, and then 2 groups of NOG-EXL mice. The NSG-SGM3 mice and one of the NOG-EXL groups were humanized in-house with busulfan preconditioning, so it's a chemical preconditioning, and then the mice received hematopoietic stem cells intraosseously. The original goal, as I mentioned, for this study was to develop an autologous model. So that was the original intention. Unfortunately, not enough cells were isolated from the patient to provide autologous cells for both of these groups of mice. And so the NOG-EXL mice received autologous cells, and the NSG-SGM3 mice received allogeneic stem cells. Our third group consisted of NOG-EXL mice, but these are the commercially available products. So they are humanized at Taconic and then delivered to us already humanized, so I don't have the details of their humanization protocol. And then this is the time course of the experiment. Mice were humanized, and then at 17 weeks post-humanization, the plan was to transplant the patient-derived xenografts intracranially into the mice, and then 5 weeks after that, the mice would be euthanized, and we performed a complete necropsy with evaluation of a full tissue set microscopically. However, at 15 to 16 weeks post-humanization, the NSG-SGM3 mice started exhibiting severe enough clinical signs that they needed to be euthanized. So they never received the tumor and did not reach the planned endpoint. In contrast, the NOG-EXL mice, all but one of them did reach the planned endpoint, which means they did receive the xenografts and completed the study as planned. And that's actually the first point I wanted to make. And I think one of the most important points from this study is that the NOG-EXL mice did survive, almost all of them, to the end of the study, allowing the study to be completed, which was not true with the NSG-SGM3 mice, both in our previous work with this model and in this study. They weren't able to survive long enough to receive the tumor and then have analyses performed on that tumor, which was the original goal of the study. And then, as I mentioned, we performed a complete necropsy with evaluation of a complete tissue set, and that included histopathological characterization, and then also at necropsy, we performed some blood works and hematology. And based on all of that, we developed a multi-parametric clinical pathological scoring system. And that gave us a composite score, like a total score of myeloid cell hyperactivation syndrome in each mouse. And that's what I'm showing you here. So these are the total scores for each individual mouse in the study. On the x-axis here, we have the different strains, NSG-SGM3 or NOG-EXL. In this graph, the two groups of NOG-EXL are combined because they're very similar. And then we have humanization status, so whether they received human hematopoietic stem cells or not. And the NOG-EXL mice in this column are affected. They do have myeloid cell hyperactivation syndrome, but it's not as severe as the NSG-SGM3 mice. And keep in mind that this is at different time points as well. These data were collected at the time of death. So the NSG-SGM3 mice, which are more severely affected, they have higher scores. And this is only 15 to 16 weeks post-humanization, whereas the NOG-EXL mice here, they're less severely affected, even though this is at 22 weeks post-humanization. So now I'm going to show you the features of this syndrome. And we're going to start with the gross pathology. These are two NSG-SGM3 mice, a control on the left and an affected mouse on the right. And the features that you might see at necropsy with mice with this syndrome include a poor body condition. So the control mouse has visceral and subcutaneous adipose stores, whereas the affected mouse really doesn't have much left. Also, the affected mouse has pallor, so this whole mouse is paler than the control mouse. They have serous blood. So in the control mouse here, you can see that the blood that has leaked out during dissection is dark red, normal consistency, whereas the blood in the affected mouse is watery and pale red. The affected mice also tend to have a small liver with an irregular surface. You can see that here. So this liver is small, and if you look closely, it has a sort of granular, irregular texture compared to this nice, smooth liver in the control mouse. And then they also have splenomegaly, typically. So this is the spleen right here in the control mouse, and then this is the spleen in the affected mouse, and it's even extending back here behind the stomach and the liver. And now I'm going to go through the microscopic features of this syndrome. So in the upcoming slides, the NSG-SGM3 mouse will be on the left, and the NOG-EXL mouse will be on the right. We're going to start with the pancreas. So in the NSG-SGM3 mice, we have the pancreatic acini here. So these are the secretory structures of the pancreas. And then in the interstitium, this is really dramatically infiltrated by a lot of mast cells. And these are human mast cells. We can tell that by the morphology and also by IHC. The inset here is CKIT. So all this round is CKIT positive cells in the interstitium. And then we confirm that they're human cells using an antibody specific to human CD45 that I'm just not showing here. But all these cells were human cells, and they're CKIT positive. So these are human mast cells. And you can see that they really infiltrated and expanded this interstitium, and then we have atrophy and loss of the normal parenchyma. Compared to the NOG-EXL mice, which have a normal pancreas. This is all normal exocrine pancreatic tissue, and there are no CKIT positive cells. And this makes sense based on the genetics of the mice, because the NSG-SGM3 mice express KIT ligand, and the NOG-EXL mice don't. Next, I'm going to show you the liver. So the liver was interesting because the overall severity of the lesions in the two strains were similar in the liver, but they had some interesting qualitative differences. So in the NSG-SGM3 mice, we have a mononuclear infiltrate in the portal regions, and it includes quite a lot of mast cells. This is a CKIT IHC again, so these brown cells are CKIT positive. And that's compared to the NOG-EXL mice, which have a difference in the distribution. These were more randomly scattered throughout the liver parenchyma, and sometimes had a central-lobular distribution. And then also the cellular characters. So these cells are more of a histiocytic to granulomatous population. In these scattered, or excuse me, in these randomly decentralobular distributed infiltrates, there really weren't any mast cells at all. Even in the portal regions, there's only a few CKIT positive cells in these NOG-EXL mice. So there's an absence of mast cell hyperplasia in the liver as well as the pancreas in these mice. And that's interesting because the pancreas mast cell infiltration was known about in NSG-SGM3 mice, but the mast cells in the liver had not been previously reported. So that's an interesting finding. And then if we look at this cellular infiltrate more in the NOG-EXL mice, as you could see even at the lower power, there's a lot of pigment here. And if we look in this higher magnification view, we have this brown pigment by color and by the sort of globular nature. It's suggestive of hemosiderin. And then within these macrophages, we have multiple erythrocytes or fragments of them. So this is consistent with hemophagocytosis. We confirmed that this is consistent with hemophagocytosis and breakdown of red blood cells by using a Prussian blue stain, so the stain's iron. And so that tells us that there's a lot of iron from blood breakdown in these macrophages. And there's less in the NSG-SGM3 mice. I have some thoughts about why that might be true. I think it might be related to the timeline of when these mice develop lesions, but I'm happy to talk about that some more with anybody who's interested at the end. Next, let's look at the bone marrow. So in the bone marrow, there's a couple things going on. First, I want to draw your attention to the insets here. So this is IHC for human CD45. So this shows us that the majority of the bone marrow is taken over by human CD45 positive cells. There's not a lot of mouse cells present anymore. And then if we look at the H&E stained photos, first I want to point out in these areas with the double dagger, and this is true on both of the strains, so the NSG-SGM3s are more affected. These are areas of increased eosinophilipoiesis, which is an excellent word, and really beautiful when you look at it in this higher magnification picture. So these are all, or almost all, eosinophil precursors. You can see the little red granules, and they're in multiple stages, immature to more mature, including a couple of nice mitotic figures. So this occurs in both of the strains, but as I mentioned, is a little bit more severe in the NSG-SGM3 mice. And then we also see aggregates of histiocytes in the bone marrow. So that's these areas with the asterisks that are a little bit more lightly eosinophilic staining. And again, this does occur in both strains, but is a little bit more severe in the NSG-SGM3. And the last thing I want to point out is that the NOG-EXL mice have a few hematopoietic cells still present in this field. These larger ones are megakaryocytes, and there's some other precursors shown here, whereas the NSG-SGM3 mice tend to have more of the bone marrow taken over and less of the normal hematopoietic cells still present. A related lesion that we saw in the NSG-SGM3 mice, some of them, but not in any of the NOG-EXL mice, at least in our study, was meningeal involvement in the spinal meninges. So I'll just orient you to these photos. This is the spinal cord. This is the meninges. This is the leptomeninges and the dura. This white space here is an artifact from processing. This is vertebral bone, and then the bone marrow. And this is normal. And in the NSG-SGM3 mice, the meninges are expanded by histiocytes, also some of those eosinophil precursors and some connective tissue. Like I mentioned, we saw this in some of the NSG-SGM3 mice, but not any of the NOG-EXLs. Another lymphoid tissue that I want to show you is the spleen. So because both of these strains are severely immunodeficient and then have been humanized, the architecture of the spleen is not normal. But what I want to point out are these aggregates of cells here and these smaller aggregates here. And these are composed of histiocytes and also lymphocytes. I didn't show the lymphocyte markers, but we did perform them. But here in the inset, this is a IHC for human CD33, so that stains our myeloid cells. And you can see an aggregate here. And then the other thing I want to point out here are these multinucleated cells, pointed out with these arrows. And then we have an inset here, higher magnification. These are also really beautiful cells, although maybe not so great for the mouse. These multinucleated cells form when macrophages are really activated. So this is evidence for activation of these macrophages. And we saw them consistently in these spleens of the NSG-SGM3 mice, but only in a few of the NOG-EXL mice. And then the last histophoto for this part of the talk, this is the skin, specifically the pinna, so the ear of the mouse. And in the NSG-SGM3 mice, in Most of the mice, we saw evidence of inflammatory infiltrates in the skin. Which you can see here, it's thickened, the dermis is expanded, and then we even have some more of those multinucleated giant cells. Whereas only very few of the NOG-EXL mice had infiltrates in the skin. Okay, I'm going to show you a couple of pieces of data from the hematology as well, starting with total leukocyte counts. So these were performed at the time of necropsy, so at the time of death or euthanasia of these mice. And here we can see that the NSG-SGM3 mice, some of them have elevated total leukocyte counts, whereas the NOG-EXL mice do not. Happy to discuss this further as well if anyone's interested. What I think might be going on here, I think it may have to do with the NSG-SGM3 mice being clinically pretty ill when they were euthanized, whereas the NOG-EXL mice were displaying clinical signs, but they weren't as sick as the NSG-SGM3 mice. And then again, keep in mind that the difference in time point here, that the NSG-SGM3 mice, these data were collected 15 to 16 weeks post-humanization, and then NOG-EXLs at 22. Another important feature of this syndrome is anemia. And that's thought to be due to a combination of the hemophagocytosis that I showed you an example of in the liver, and then also because the bone marrow is taken over by the eosinophilophysis and somewhat the histiocytes, and there's not a lot of normal erythroid lineage development going on. And so this is pretty similarly severe in the two strains, at least at the time of death. So these are our non-humanized controls up here. Not sure what happened with this one. And both strains that were humanized had a pretty severe anemia. But again, like I mentioned on the previous slide, these are at different time points. So this was at 15, 16 weeks post-immunization, and this was at 22. So in summary of the features of myeloid cell hyperactivation syndrome and how each of these strains are affected, we can see that for most of the features, the NSG-SGM3 mice are more affected, except for the anemia and the hemophagocytosis, where the two strains are pretty similar. And this altogether leads to a longer survival in the NOG-EXL mice. And so we concluded that if you need to do a study that requires long-term survival post-humanization, you have a longer window in which to complete your studies with the NOG-EXL mice than the NSG-SGM3 mice, and so they might be a better option for your study. So that's really the main takeaway. In the next few slides, I want to talk about a couple more interesting features that you may have questions about, and that I think, like I said, are interesting. And so to start that off, I just want to remind you of our experimental design, because we had this patient-derived xenograft transplantation at 17 weeks post-humanization. So the NOG-EXL mice were able to undergo this transplantation, the NSG-SGM3 mice weren't. And so we were wondering, as we were looking at these data, is there any effect of the xenografts on, or any association with differences in severity of the myeloid cell hyperactivation syndrome with the xenograft? Turns out there's not. Here I am showing the NOG-EXL mice. Now in this slide, they're separated into whether they were humanized in-house or were the commercially available product humanized before we got them. And there's no difference in either group based on the presence of detectable tumor in the mice. And then another thing that you might be wondering about, and that I also want to discuss, is the hematopoietic stem cell engraftment in these mice. If you're using humanized mice, that's always a big question, right? How well did they engraft? So first I want to talk about, excuse me, the peripheral blood population. So these are circulating cells. On the left here, these are the lymphoid cells, and this was done by flow cytometry at 76 days post-immunization, so about 10 weeks. And we only have data from the mice humanized in-house. The NSG-SGM3 and the NOG-EXL is humanized in-house. And at this time point, the lymphoid population is pretty similar in the two strains. The myeloid population in the peripheral blood, at least, so these are the circulating cells, is a lot lower in the NOG-EXL mice, which is interesting, given that they have a less severe myeloid cell hyperactivation syndrome. So we can, again, discuss this a little bit more if people are interested, but I have a little bit more data to talk about. So I just want to point out that the data I showed you initially on this slide was 76 days post-humanization. So this is the last time point that we had flow data from for the NSG-SGM3 mice, but the NOG-EXL mice, because they lived longer, we had another time point. So this was 123 days post-humanization, which is about 17 weeks, I believe. And at that time point, they did have a similar number of myeloid cells circulating to what we had seen in the NSG-SGM3 mice at an earlier time point. And then the other component of this that I want to talk about is what's going on in the tissues. Because looking at the circulating cells is certainly important and it is one component, but tissues are a different compartment. In fact, multiple different compartments. So I'm going to show you some IHC that we did for immune cells in the tissues. First, we're going to look at the lymphocyte populations. So we did IHC for T cells with CD3, and B cells using PAX5 as markers. And these are lymph nodes in these mice. And for both strains, the lymph nodes are populated by both T and B cells. Again, the architecture is not normal because these are such immune deficient mice that have been reconstituted with human immune cells. So they do go to the lymph nodes, but it's not a normal architecture, but they are present in both of these strains in the lymph nodes. I'm not going to show you the images, but the spleen similarly had both B and T cells in both strains, although there were a mildly higher percentage of T cells in the spleen in the NSG-SGM3 mice. And then I want to show you the myeloid cells in the tissues as well. We're going to start with the lymphoid organ, so lymph node and spleen. In the lymph nodes, we do have some myeloid cells, mostly around the periphery. In both of these mice, looks pretty similar. The spleen I showed you earlier, where we have some of these aggregates that are more common in the NSG-SGM3 mice. And then we can see that those multinucleated cells are myeloid cells, myeloid origin. They're not megakaryocytes because they are positive for CD33 as well. The NOG-EXL mice have fewer of those aggregates and fewer of those multinucleated giant cells, but they do have myeloid cells present in the spleen as well. And then in some non-lymphoid tissues, we did look at the liver and the pancreas earlier with the H&E, but here with IHC for myeloid cells, we see a good population of myeloid cells in the liver. In both strains, the staining quality is a little bit different because the cell's morphology is different in these two strains, but these are all myeloid cells, which makes sense. We saw they were macrophages earlier by H&E. We saw that there were some mast cells, so those are all myeloid cells. So we know that they are present in the tissues. And then in the pancreas, because we have so many mast cells, those are myeloid cells. So we see that there's a whole bunch of them in the pancreas of the NSG-SGM3 mice, and not a lot going on in the NOG-EXL mice. So it's a little bit of a little more complicated picture as far as when and where the myeloid cells are in these mice. Like I mentioned earlier, I think there may be differences in the timing of the development of the syndrome, and especially because of the presence of KIT ligand in the NSG-SGM3 mice, differences in the tissue distribution as well. But I did, I'm not showing the data, but I did look for an association between the myeloid cell hyperactivation syndrome score in the individual mice and their percentage of myeloid cells in the peripheral blood, and there was no association in this study, at least. So I suspect that there is that the tissue distribution may be more important than what's going on in the peripheral blood. So in conclusion, the NOG-EXL mice do exhibit myeloid cell hyperactivation syndrome, but it's less severe than the NSG-SGM3 mice, and you have a longer window to perform studies that require long-term survival post-humanization. The NOG-EXL mice in their myeloid cell hyperactivation syndrome are overall similar, but there are some differences from the NSG-SGM3 mice. Most notably, there is no mast cell hyperplasia in the NOG-EXLs. Like I mentioned, the tissue distribution is less extensive. They do have a similar anemia, and like I said, a longer survival. At the end here, I just want to mention some of the outstanding questions that are sort of interesting to think about in this work. A big one is the mechanism of this syndrome and why there are strain differences. I've mentioned KIT ligand a couple of times. I think that makes a lot of sense for the differences in the mast cell hyperplasia in these two strains. There's also evidence that the NOG-EXL mice have a lower level of cytokine expression, so the GM-CSF and IL-3, than the NSG-SGM3 mice. They are still higher than non-myeloid supportive strains, but not to such a high level as the NSG-SGM3 mice, so that's probably related to the difference in severity and probably also the difference in timing. I mentioned briefly that the NSG-SGM3 mice had some more T cells in the spleen. That was true also in the liver. And so I'm curious whether those have a role in the more severe manifestation of the syndrome in that strain. There's some previous work from the Comparative Pathology Core on the NSG-SGM3 mice didn't find a lot of T cells present in those lesions, so that's a difference in this study, and I think that's interesting. I also think it's interesting because the human condition of hemophagocytic lymphohistiocytosis, which shares some features with this, does have an involvement of T cells, so unclear how they might be involved here. It is possible that there's some variability among donors. The NSG-SGM3 mice, this syndrome has been demonstrated pretty consistently across donors, across experimental settings at other institutions, so I think it's pretty characteristic. The NOG-EXL mice, we only had one donor in this study, but I have seen it in other studies, and I have heard from other pathologists that they've also seen it in their mice at other institutions. So while there's certainly a possibility of that contributing, again, I think it's more than just different donors. I mentioned that I think there may be something interesting going on, thinking about which cells are in the tissues versus which cells are circulating, especially because we're thinking about myeloid cells. And then I mentioned the timeline a couple of times. So we see that the NOG-EXL mice exhibit clinical signs later. Those liver lesions, I think, may be qualitatively different in the different strains because we might be catching them at different times in their development of the lesions. So I think it would be probably interesting to look earlier in these mice. And I know one of the questions submitted by or during registration was about sort of earlier sort of preclinical markers, what could be monitored before the mice develop these more severe clinical signs. And I think to look at that, I think probably doing some experiments where they are evaluated earlier in the presence of disease, not just earlier in the timeline post-humanization, whether there are certain aspects of the syndrome that develop earlier than others, I think would be pretty interesting to look at. And that goes to my next point here, whether there are differences before the clinical signs. Maybe they start developing anemia early. Maybe they start losing a little bit of weight. Maybe they, decrease their food intake. Maybe the cytokines are different earlier on, and that could be monitored, and we just don't know that yet. And then the last point that I think would be interesting is whether this affects other mouse strains. I have heard anecdotal evidence that Mr. G6 mice have something similar to this going on, although I have not worked on those myself, but I think that would also be quite interesting. So at the end, I just want to say that this work was done by a great group of people. I am the representative here, but lots of people were involved in this, and I want to thank them.